Welcome you all to the Mana Lab biology session. So today we'll take up a very interesting topic. Let's see what we are going to study today. What do you see here? Can you see anything? Huh? Tell me what you all can see. See, you can see the beautiful flowers, no? You can see the beautiful flowers over here. Clear? So, see, you can see here the beautiful flowers over here. See the flowers over here, beautiful color flowers over here. You can see the red flowers over here. You can see colorful white and red over here. Colorful flowers. So colorful they are. Fantastic, no? The world of flowers. So what are these flowers blooming? Where do you get these beautiful flowers from? No, from the trees. So these flowers are born on the tree, no? And plants, etc. When do you get these flowers? See, these flowers, they are the major specialized short shoot, They are modified leaves. The leaves are known as sporophylls. The leaves are known as sporophyll, okay? Now, in this sporophyll, what do you get? See, flowers. The sporophyll gets modified into flowers. Clear? Now, these flowers, they are not only beautiful and reproductive part of the plant. See, they are used for making various types of dyes. They're used beautiful, colorful dyes you get from these flowers. And you get beautiful perfume scent from the flowers, beautiful scent you get from the flowers. You see, these are the food coloring agents. The food coloring products also we get from the flower, Besides this, not only the dyes for clothes, food coloring agent, the perfumes and scents you get from this flower. Besides that, we are also used for a cultural fest, various cultural products. And even for religious products, we use this. For various religious flowers also, these flowers are used. See, as a decorative purpose. So you can see how beautiful the flowers are. In so many ways they are utilized, They are not only the sexual reproductive part of the plant. The flowers have got various aesthetic value also. When we are depressed, go to a park and see the beautiful flowers around you, How beautiful. Just imagine yourself walking here in between these plants, When you just imagine if you're walking over here with these beautiful flowers over here, or you're using any of this product over there, just imagine how nice it is, How nice it is. So now let's go these flowers as the major reproductive part of the plant. Besides its usefulness, how they are produced. So let's know how these flowers help in the reproduction. Okay, so now we'll next move on to know more about this. So now today we'll be starting the chapter. Why I was talking about flowers, you may be wondering, no? See, flowers are the most important part of the plant. They are the most attractive, you can see, so attractive they are. So they are not only attractive part of it, and they've got so many useful. Besides that, we are going to talk about the sexual reproduction in plants. Flowering plants that to angiosperms and all. So here the sexual reproduction is brought about by what? From these flowers only, So today let us do flowers as the major part where it takes an important role in the sexual reproduction in the flowering plant. Okay, so now today next part we will just take up and see how these flowers help in sexual reproduction. So now we know flowers are not only the object of ornamental aesthetic value, they're excellent offering to God in cultural fest, etc. They're used in our Hindu rituals, etc. The beautiful colors, the scent we get from the flowers, they affect the human mood. Not only the sight, The scent also affects our mood a lot. So there will be a source of inspiration for us. Not only for us, even to the great poets, when they see these beautiful flowers, words just flow out through the mind and they put it in flower words. So flowers are also used for scent, colors, etc. Now let us see. Flower serves as one of the important tools in identification of a plant also, When you look at a flower, you will know what type of plant it is, what plant it is. You can tell the name. No? Easily you tell the name. Just you see if the flowers are not there, it is quite a bit difficult for us to know. But if presence of flowers, so easily that will help us in identification of the plant. It also will help us in establishing the relationship of various groups in the angiosperms. So such is the flower. So flower, the specialized sporophyll, how it takes an active role in the reproduction. Let us see. So sexual reproduction in flowering plant. So now you see, these flowers always develop from the floral bud. We know very well, a floral bud gives rise to a flower. So if these are the bud, how are these floral buds going to get converted and form a flower? What are the important things that is mainly necessary for it? To bring about a change in the structure of the floral bud into a flower. So the structure from floral bud is converted to a flower for you. So it means in the plant, it first appears as a bud. Now this bud gradually blooms, opens up to form a beautiful flower for you. To form a beautiful flower for you, it opens up. So what are the things that is mainly important that helps you in controlling, especially hormones? Hormones play a very important role for you to bring about the structural morphological change of the floral bud into a beautiful flower. So it means the actual decision to develop a flower will be taken much before it appears, much before it appears of the bud also. It has already been decided. And what is that has decided? So it is a vegetative apex which will change into a floral apex. See, this is the floral apex. This is the floral apex. The vegetative apex has got converted into a floral apex. And now this becomes flat. It's slightly, once it gets converted to floral apex, the tip becomes slightly fat. Flat it becomes. The apex of it becomes flattened. Why? Various floral organs has to develop on this apex, That is why it becomes flat. So you can call this as primordial thalamus, where it is going to become future thalamus. And from towards the center, gradually, the hormonal activity takes place internally. And slowly, from the flattened apex, gradually the floral bud makes its appearance. And from there, step by step, the different orals take its origin, and then it forms finally the flower. And then it forms finally the flower. So to bring about the structural change in it, we require the presence of this hormonal activity. The hormonal activity is very much important to bring about a structural difference and convert the floral bud into a flower. Okay. Now, once these flowers are formed, how beautiful it is. Now, let us see the structure of a flower especially. See, this is with reference to the structure of a flower. What are the different parts that we get in a flower? Flower usually consists of 4 whorls. Now you can see the first, this is the pedicel, what you call it as a stalk of a flower. The stalk of a flower, it takes its origin at the tip of the floral axis, and you can see the articulation on either side to form the floral axis, and gradually it slightly flattens. This flattened part is now referred to as the thalamus. This flattened part is now referred to as the thalamus. So now this thalamus forms a stage flat structure where the four orals takes its origin. The first oral, you can see this green structure is the calyx. Each and every green part is known as sepal individually. Collectively, we call them as calyx. Collectively, they are referred to as calyx. This forms the first oral. This forms the first oral on the thalamus or receptacle. Next forms the most beautiful part of the flower. That is this beautiful colored petals. Each one is known as a petal, and together it is known as the corolla. The next oral towards the center, that is when you move from the marginal towards the center, the next one is the colorful part, the petal. And collectively they are known as corolla. This forms the second oral. This is the second oral in the plant. Okay, so the second oral forms, the first is the calyx, the second one is the corolla. Now coming to the third one. Now sepals are usually, you can see before coming, the sepals are usually green in color. You can see they are green in color. And the function is to protect the inner parts. Usually the sepals, their major function is protection. The major function is protection. Especially when the flower is in the form of a bud, they protect. So whereas the petals are the most colorful part and they form the most attractive part of the flower. So calyx and corolla, these two parts, which we now studied. See, this is the calyx, the green ones, which are protective in nature, forming this part of it, and corolla, the attractive, colorful part, the attractive part of the plant, this one. These two, the calyx and corolla, these two are not involved in the mechanism of reproduction. So we call these two as the accessory part. The calyx and corolla is referred to as the accessory part, or you can call them as the accessory orals in a flower, because they are not directly involved in a mechanism of reproduction. They are known as the accessory orals in a plant, the calyx and corolla. Next coming to, see, next we are going to talk about the essential parts. So now we are going to now talk about the essential part in a flower. So when we talk about essential, definitely it has to be the reproductive part, which is the major reproductive part of a plant. So in that, first let us see here the anther. The anther is the male reproductive part of a plant. The male reproductive part of a plant is the anther. So let's move back over there. So now you can see here, the male reproductive part of a plant is the anther. Clear? So this forms the major male reproductive part of a plant. Okay. Now, this male reproductive plant, what do they contain? See, they contain a filament, an anther at the top. This totally is referred to as microsporangium. This is the filament. This is the anther. Together, it is called as a stamen. Together, you can see here. Together, it is called as a stamen. And this forms the major reproductive or the male reproductive part of the plant known as microsporangium. The anthers are the microsporangium. So if this is the male, let's go on to the female reproductive part. So now coming to the female reproductive part. Now see, this part forms the female reproductive part that is known as the carpel, or it is also known as the pistil. It mainly consists of an ovary. The swollen part is the ovary. The long narrow neck is the style, and the lobe structure at the top, slightly swollen one, is the stigma. Just like this consists of a filament, the male one, and this is the anther. So the male consists of the filament and anther, together forms the stamen, the male reproductive part of the plant, and while the female carpel or pistil consists of the swollen ovary, a long narrow neck style, and the tip swollen part is the stigma. So the stamen and the carpel are the reproductive part in a flower. That is the third whorl, the stamen, and the fourth whorl, the carpel. The third and fourth whorl in a plant, that is in a flower, forms the reproductive part, which is the essential part in a flower. Okay. Now let's move on to see individually. The stamens are known as microsporangium, and the carpel is known as megasporangium. The carpel is known as megasporangium, and stamen is known as microsporangium. So now coming to, let us go one by one. How does the development of microsporangium takes place and the megasporangium takes place, and to what does it lead to? Clear? Let's move on to the next one. See now we are going to talk about the male one. That is, male is what? We are going to deal with the stamens. That is the stamen. Now we know very well, stamen is the male reproductive part. It consists of a filament. You can see here, this is a filament. And at the tip part, this is the anther. So together it is called as a stamen. Do we find, so here what do you find in this? See, this filament is of different height. See, this is of this length, this is of this height, this is of this height, this is of this height, this is of this height. So it means, it tells that the filaments are not of the same height in a flower. It varies. See, they are different heights in a flower. So this different height in a flower condition, such a condition is commonly referred to as an heterostyle condition. Is commonly referred to as an heterostyle condition. So you can see here the different height of the filament in this flower shows the heterostyle condition of a flower, which tells that hetero means different, style means the length. So the style is of different length over here. So this shows the heterostylic condition. What is the advantage of this heterostyle condition in some flowers? In case of some flowers, you know, this heterostyle condition helps them in pollination and other processes. So that's why this is of a heterostyle condition. Okay. Now let us go on to the next part of it. So you can see this is one height. Height varies. The tall white, these are slightly shorter than this, seems to be the tallest one. And you can see of different, this is shorter one. So it means hetero means what? Different height. The presence of filament in different height tells that is heterostylic condition. Now let us see. It means this filament with the anther, totally the stamen, varies from one flower to another flower, from species to species. The length varies. The difference in length is known as heterostyle condition. Now let's move on to talk about this stamen. Now you see, stamen mainly consists of what? It consists of a long, thin, elongated structure. It consists of a long, slender stalk. So this is a long, slender stalk. The filament is nothing but a long, slender stalk. It is a long, slender stalk. Okay, so the filament is nothing but a long slender stalk. And what does this filament mainly consists of? Now, this long slender stalk, the filament, which will terminate in an enlarged part. The terminal part of that enlarged part, let's see what it contains. Yes, you can see here. The terminal part, the enlarged part over here, mainly consists of a lobed structure, and this is known as anther. So this is a slender stalk. And this lobe structure at the top, what it bears, it terminates into an enlarged portion, what is known as anther. All stamens collectively, and this anther, it has got a two partition. You can see a partition in the center here. So that part, it is two lobes here, and the center here. The center one is known as a connective because it has got two lobes on either side. A typical angiosperm, this is known as bilobed. This is known as bilobed. Okay, so the anther is bilobed. The anther is bilobed. Bilobed means 1 and 2. It consists of two lobes. It consists of two lobes, so it is known as bilobed anther. And in the center, it is connected by the connective. In the center, it is connected by the connective. Clear? So it means the slender stalk has a swollen region at the top, and the swollen region is known as anther. This anther is having two. The anther is a bilobed one. It has two lobes. So 1 and 2, two lobes. So it is bilobed. And this is also, each lobe is having intern two theca. Each lobe is having intern two theca. That is this one lobe, and this one lobe is having two theca. So we call it as dithecous. So it is dithecous. So this bilobed, one minute. It is bilobe structure and dithecous. Bilobed and dithecous structure. So this longitudinal groove, what is connecting on lengthwise is the connective, is majorly the connective part of it. So all stamens totally, and let's see this connective in turn, what does it bear, or what does this anther mainly contain of? So usually mainly this anther, totally or collectively we call this as androecium. We call this as anther or androecium. So it is the male reproductive oral or reproductive part of a flower. And what does this anther usually produce? If it is the male reproductive part, what does it produce? See, the anthers mainly contain, like told, see, inside this anther, it has got chambers. Each side, it will be one here, one here, one here, one here. Each one is known as pollen sac. So on each anther, on each lobe is having two sacs. That is why we call it as dithecous. Dithecous. And what is present? These are known as pollen chamber or pollen sac. And what is present in this pollen sac? It is in this pollen sac, the male functional unit of the stamen produces known as the pollen grain. Pollen grain. Okay. So it means it is a slender filament. The filament is slender and long. At the top, it consists of a swollen structure known as anther. The anther is having how many? Two lobes. It has two lobes. That is why you call it as what? Dithecous. That is why you call it as dithecous, two lobes, dithecous. And also, what is the other name? Because bilobed. You call it as dithecous, bilobed anther. Bilobed and dithecous anther. And what is present inside this? What do they contain? Being the male reproductive part, we also call this as androecium. And what do they produce? They produce the functional unit of the male reproductive part, that is the pollen grain. So that is the pollen grain. Okay, so this each thecis consists of this pollen sac over here, and they contain the pollen grain. Pollen grains are produced inside this pollen sac. Pollen grains are produced inside this pollen sac. Okay, so now this dithecus, you see, it is actually bifurcating it. Suppose if you take a cross section of this anther, this is what we see inside. When you take a cross section of this anther, internally you can see here, you see, internally you can see the presence of pollen sac 1, 2, 3, 4. So in each lobe you can find 2 pollen sac. Each lobe you can find 2 pollen sacs. So 1 over here, 1 over here, 1 over here, 1 over here. So each lobe you can find 2 pollen sac, and inside this pollen sac, what are produced? The pollen grains are produced. It is the pollen grains are produced inside this pollen sac. So this is the male reproductive part, the filament with the anther, which is two-lobed or bilobed, dithecous with a connective in the center. When you take a section, has presence of pollen sacs, and inside this pollen sac, you find the production of pollen grain. So let's move on to the next one. So when you if you just take a transverse section to know totally internal structure of the anther. So let us now know the internal structure, that is transverse section of an anther. So this is the transverse section of an anther. So in this transverse section of an anther, what are the major structure it is having? See, it is made up of an outer layer, you see. It is in the transverse section of an anther, that is the male reproductive part. You can see here, it is made up of outermost layer is epidermis. So epidermis is the outermost layer. Clear? Epidermis is outermost layer, and this outermost layer is highly protective in nature. So epidermis is the outermost layer and it is protective. What is its function? Protective. This forms the outermost layer. So immediately below the epidermis you have the endothecium, the middle layer. So next, followed by the endothecium, it is next followed by the endothecium, and internal to that you have the middle layer. So this is the endothecium, next is the endothecium, and finally you have the middle layer. So this epidermis, endothecium, and finally the middle. Then internal to this, the innermost layer, you can see, and one of the important layer here, it lays here, that is the tapetum. The elongated cells here form this tapetum. So let us now see the outermost layer. This is the outermost layer, epidermis. This epidermis is the outermost layer. It is only one cell in thickness. You can find this outermost layer, epidermis, is just one cell in thickness. So these are the one cell in thickness. You can find here the outermost layer, epidermis. You can see here what you're seeing here. See, throughout you can find here, see the single layer here, barrel shaped cells. You can see here these cells forming the outermost layer, which is a single layer cells. This forms the epidermis. So this is outermost layer and it is just one cell in thickness. It is just one cell in thickness. You can see in the center how it is curved, how it is curved here in the center. And this region you find here, this region is known as stomium. This region is known as stomium. Clear? This region is known as stomium. So you can see here, it is a single layer of cells now forming the epidermis. So this is a single layer of cells, outermost layer forming the epidermis. Okay, which is highly protected. Now immediately inside this, what is the next layer? Endothelium. This layer is present just below the epidermis. You can see here, this layer just below the epidermis. This layer is known as the endothelium. This layer is known as the endothelium. It is also a single layer, and you can find the cells are radially arranged. See here, this is also single layer, and the cells here are radially arranged. Okay, so immediately below that you find radially arranged cells forming the endothelium. Radially arranged cells forming the endothelium. So here, these cells, they have fibrous thickening. These endothelium cells, they have a fibrous thickening. The cells in this endothelium, they have fibrous thickening. Okay. So the cells of endothelium, they have fibrous thickening. Clear? So this fibrous thickening, they help during the mechanism dehiscence. Dehiscence means when this is going to, on maturation, it is going to split and release the pollen grains. No, so this fibrous thickening will help in dehiscence. Okay, this will help in dehiscence. It will help in dehiscence of what? Dehiscence of the anther on maturation. So in between these cells, few cells are without thickening. In between, there are few cells, that is here in this region and here in between. Few of the cells here located in this region and this region, few of the cells do not have this fibrous thickening. So that region is known as stomium. This region where the few endothelial cells do not have the fibrous thickening, these cells are known as stomium. So this forms the second layer. This forms the second layer. So it means stomium does not have fibrous thickening. Clear? So stomium cells, or the cells present here, do not have fibrous thickening. Which one? The cells present in the stomium do not have the presence of fibrous thickening. Then internal to this, so that forms the second layer. Now comes the middle layer. So now you can see this innermost layer, this just below this cells, radial arranged cell, the innermost cells are known as the middle layer. This forms the middle layer. So what is this? This middle layer, usually there are presence of 3 to 4 layers present just below the endothelium, nearly 3 to 4 layers. This middle layers may contain 3 to 4 layers of cells. They contain nearly 3 to 4 layers of cells just below the endothelium, and these are known as the middle layer. These layers are what happens means they usually disintegrate by the time the anther matures. When the anther undergoes maturation, this middle layers, present throughout here will undergo disintegration. They undergo disintegration. They generally disintegrate. What happens to them on maturation? They undergo disintegrate on maturation of the anther. On maturation of the anther. So they undergo disintegration on maturation of the anther. So why do they undergo disintegration? This disintegration of the cells help in the dehiscence of the anther. Okay, this disintegration makes way for the mechanism of dehiscence of anther. So once the anther undergoes dehiscence, it helps in releasing of matured pollen grain. It helps in releasing of matured pollen grain. Okay, so it means few cells are present, 3 to 4 layer of cells are present. This forms the middle layer. They are simple cells. Once this anther undergoes maturation, they undergo disintegration. The disintegration of these cells helps in the mechanism of dehiscence of anther, thereby releasing the matured pollen grain. Next, come to the innermost layer. This longitudinal cells, what you find here, this is the innermost layer here. This layer is known as tapetum. This innermost layer that is present is known as tapetum. What is this tapetum? This is the innermost layer of the wall. This is multinucleated. You can see here. They have presence of many nucleus. So this is tapetum characteristics, multinucleated. They have the presence of multinucleus, more than a single nucleus. Number one, they have dense cytoplasm. They have the presence of dense cytoplasm. And if they are multinucleate and dense cytoplasm, what is the function? They are supply nutrition. They're mainly responsible for supplying nutrition. They supply nutrition to the growing of the microspores. So tapetum secretes a type of enzyme and also hormones. They not only supply nutrition, they also secretory in function because they secrete certain enzymes and also hormones. So they are secretory in function, at the same time nutritive in function. So they have the presence of glandular cells in the tapetum, which secretes enzyme. They also have these amoeboid cells. These amoeboid cells will get separated and they move freely in the pollen chain. So this forms the major 4 layer. Then coming to the internal region, this 4 regions forms the, this is the pollen chamber. So this innermost region, the cavity, the central cavity of the anther lobe is this pollen chamber. It is here the microsporogenesis takes place. It is over here, the it is the site, it is the region where the mechanism of microsporogenesis takes place. So how does this mechanism, microsporogenesis takes place, or how does this process of microspore mother cells or microsporogenesis takes place? So let's move on to see how that process takes place. Before that, let's come back once again to see what does this. See, these are the outermost layer, like I told you, single layer, protective, endothecium, single layer. Endothecium is also single layer, fibrous thickening, cells, and middle layer, 3 to 4 layers. 3 to 4 layers. This usually disintegrate on the maturation of the anther. You can see an innermost layer. You can see multinucleated this cells, the tapetum. which are nutritive, secretory, secreting enzymes, as well as hormones. This forms the four important layers in the case of. So you see, epidermis, endothelium, middle layer, and innermost layer is called as the total one, and the innermost layer is called as the tapetum. This layer provides nourishment for the development of the pollen grain in the pollen chamber. And inside this, there's the presence of four pollen chamber. And in the core of this four pollen chamber, we have the microsporogenesis, that is microsporangium, which contains the sporogenous tissue here. This contains the sporogenous tissue, and it is this sporogenous tissue called as the pollen mother cell, which is called as the PMC or the pollen mother cell. It is this pollen mother cell undergoes microsporogenesis and develops into a pollen grain. So where does the mechanism of microsporogenesis takes place? That is in the pollen sac here. It is here. And which is the initial stage, the first cell that starts for the mechanism is the pollen mother cell. The mechanism of microsporogenesis commences with the PMC or the pollen mother cell. It is this pollen mother cell that undergoes division and finally result in the mechanism of production of a fully matured pollen grain. So this process of development of the anther, the sporogenous tissue with meiotic division, and finally forming the pollen grain is known as microsporogenesis. So microsporogenesis commences from the pollen mother cell. Clear? Now let us take, how does this mechanism of takes place? See, now coming to the microsporogenesis. This microspore mother cells, it starts with the PMC or the pollen mother cell. It starts with the pollen mother cell. So these pollen mother cells are diploid in nature. They have 2n number of chromosomes. They have 2n number of chromosomes. Okay, these pollen mother cells, they are diploid and they are 2n in number. That is diploid in number, 2n number of chromosomes. So now this 2n number of chromosomes, these pollen mother cells, they undergo repeatedly. Usually a young anther in the beginning, it all starts with a parent gametous cells, and few of the parent gametous cells, gradually they form four-lobe structure and forms the hypodermal, and few of the cells next get converted into the primary parietal cells and primary sporogenous cells in the inside. So now this primary parietal cells and primary sporogenous cells, they now divide. They now divide, forming the primary pollen cell. So now this microspore, which is diploid, undergoes repeated mitosis cell division. Undergoes repeated mitotic cell division. Now this primary parietal cells, that is endothelium, middle layer, and tapetum. See, coming back to this, let's start one thing. The primary parietal cells will divide to form 3 to 4 cells. So it means the outermost cell will form the epidermis. If you go back to the previous structure, so the outermost cell, you see, the outermost cells forms the epidermis here. Outermost cell, and the primary cells, that is the archiporial or archisporial initials, will divide to form parietal cell and inner sporogenous cell. See, the outer parietal cell, the outer parietal cell will now divide to form number 1. It will divide to form number 1, endothelium, and second one, the middle layer. endothecium, and second one is the middle layer. So this primary parietal cell will now divide and form the endothecium and the middle layer. Then what about the internal cells? Along with that, it will finally result also in the formation of tapetum. Now, the primary sporogenous tissues are present inside. These three develop from the outer parietal cells, and the inner sporogenous tissues will divide and redivide to form a mass of sporogenous cells. They form a mass of sporogenous cells. So it is this mass of sporogenous cells will now start functioning as the microspore mother cell or pollen mother cell. It is this mass of sporogenous tissue which will now start functioning as the microspore mother cell. So what happens to this microspore mother cell or pollen mother cell? So now this pollen mother cells or microspore mother cell is diploid in nature. So development of this pollen grain from the microspore mother cells means development of the pollen grain from the primary pollen mother cell into pollen grain, matured pollen grain, is known as microsporogenesis, is referred to as microsporogenesis. So it means it is a process of formation of microspores or pollen grains. From where? From the pollen mother cell. And what is the mechanism? Means a pollen mother cell, it commences its stage from a diploid to a number. So being diploid, it gets converted into a haploid matured pollen grain. So to get converted into a haploid matured pollen grain, it has to undergo meiosis, that is the reduction division. So this development or the process of formation of microspore or the pollen from the pollen mother cell by the mechanism of meiosis is known as microsporogenesis, is known as microsporogenesis. Now this process in which each microspore mother cell will divide meiotically to form 4 haploids. See, each cell will divide to form 4 haploid. Each of the cell is haploid. They have only n number of chromosomes. They divide to result in the formation of 4 haploid microspores or 4 haploid pollen grains. The arrangement of these pollen grains varies. From one plant to another plant, it varies. But how does the formation takes place? So this microspore of pollen grain is the first cell of the male gametophyte. Microspore pollen grain is the first cell in the male gametophyte. So what happens now? This pollen mother cell will undergo meiosis, which is diploid in number, will undergo meiosis and result in the formation of a 2 haploid cell. See, a single cell has now divided and resulted in the formation of a 2 haploid cell. So this is the reduction division where diploid is converted to haploid. So that forms the first meiotic division. It results in the first meiotic division where the cell produces 2 haploid cells. So it means in this, once it undergoes, this again enters into a second meiotic division where the 2 haploid will again divide into, resulting in the formation of a 4 haploid nuclei. 4 haploid nuclei. So cytokinesis follows at the end of meiosis 1, followed by meiosis 2, and results in the formation of 4 microspores. And the arrangement of these microspores, because 4 are formed, it is known as tetrad, and the arrangement varies from one species to another. Either they may be arranged in tetrahedral or in a linear form or in a T shape. Either they can be arranged in a T shape like this, or they can be arranged one below the other in a linear form, or they can be arranged in an isobilateral like this. So the arrangement of this varies from one species of plant to another species of plant. From species, but the second meiotic division results in the formation of 4 pollen grains. These 4 pollen grains are haploid in nature, which are resulted from the production or which are formed from the pollen mother cell. So this sporogenous tissue formation giving rise to the tetrad is known as microsporogenesis and each one develops into a haploid pollen grain, and this haploid pollen grains are present inside the microsporangium, and like this numbers of haploid microspores are present, or the pollen grains are present inside the pollen chamber, which are produced during the microsporogenesis. So once the microsporogenesis completes, you find in the resultant of a large number of spores. So you can see here how the microspore nuclei will now undergo division on maturation, the first mitosis, and it will divide into two cells. One is known as the generative cell, another one is known as the vegetative cell. One is known as the generative cell, another one is known as the vegetative cell. So this generative nucleus and vegetative nucleus, the generative nucleus and the vegetative nucleus are the bicellular polar structure. It once again enters into a second mitosis, during which only the generative cells will divide into two male gametes. So here you find one vegetative and two male gametes. It will be at the 3-cellular stage or 3-nuclear stage. It is said to be fully matured. It is said to be the fully matured pollen grain. It is said to be the matured pollen grain. So it means the microspore haploid cell will undergo maturation by the formation of a first mitosis, during which it will divide into one generative nucleus or generative cell and another one vegetative nucleus. Now that ends the first mitosis cell, which forms the bicellular pollen. So this is a 2-cell stage. 2-cell stage. In some plants, at this 2-cell stage, pollination takes place. But in majority of the angiosperm, it enters into a second mitosis division, during which the generative cell will once again divide and result in the formation of a 2-male gamete. You can see it result in the formation of a 2-male gamete. So that is a tricellular pollen. It contains one vegetative nucleus and two male gamete, which are nothing but haploid male nuclei. Haploid. So this tricellular, so it is now ready for the mechanism of dehiscence and to be released. Now let's see the matured pollen grain, what it contains. Now coming to a fully grown matured pollen grain. A fully grown matured pollen grain consists of an outer layer known as the exine. It consists of an outer layer known as the exine. This exine is made up of a chemical substances known as sporopollenin. This exine is made up of a chemical substances known as sporopollenin. The sporopollenin is said to be one of the most resistant organic material that exists on this earth. Most resistant. It is said to be the biological most resistant substances which can withstand high temperature, strong acid and alkali nature. And this exine usually has certain ornamentation outside. Certain ornamentation in between, you can see small gaps. These gaps are known as germ pore. These gaps are known as germ pore. So these gaps form the germ pore. It is from this germ pore you find the pollen tube coming out later on germination. The pollen tube exits from this germ pore. So this hexanin made up of sporopollenin, which is biologically the most resistant substance which can withstand very high temperature. Any type of pH value, let it be acidic or alkali. It can withstand very acidic. And the organic substance is also not affected. No enzymes can easily dissolve it. This organic substance, there's no enzymes effect on this. So it is because of this reason only these pollen grains can be easily preserved as fossils. They can easily be preserved as fossils. Now coming to the inner layer, this inner layer is known as intine. This is very thin and uniform. And what is it made up of? It is mainly made up of pectocellulose. It is mainly made up of pectocellulose. At the time of pollen germination, the intine will comes out of the germ pore in the form of a pollen tube. So this intine will come out of the germ pore in the form of a pollen tube. So usually when this flowers, we can see this is a generative nucleus, this is a pollen tube nucleus. So this in turn, along with the intine, comes out in the form of a pollen tube. So especially in those flowers where it is insect pollination, the exine of the pollen grain will be covered with yellow, thick, viscous, sticky substance, and that sticky substance is the pollen kit. In insect pollinated flowers, this exine pollen grain will be covered with a thick yellowish sticky substance known as pollen kit. Maybe this is perhaps the protective envelope which also will stick to the abdomen of the insect and help in the mechanism of pollination. Now the cytoplasm of the pollen grain is very rich in nutrient here. This is a very rich in nutrient. It has got a lot of nutrients here. In many parts of the world, pollen tablets in the form of tablets, in the form of syrups, they are taken, as a supplement, as a protein supplement, these are taken. So, and the size and the shape of this pollen grain, especially the exine, which I told ornamental, just by looking at the ornamental, some taxonomy can tell to which family this pollen grain belongs. So that branch of science in which we can just look at the pollen grain, know the character of the pollen grain and study is known as palynology. So studying of pollen grain specialization is known as palynology. So a person who is specialized in this palynologist, just by looking at the ornamental type of the exine, he is able to guess this pollen grain belongs to which species. So let us see if this is supplement also. Let us see in what way this is useful also. See, pollen grains, they have got harmful effect as well as useful effect. See, they cause asthmatic symptoms. They are highly nasal allergic. You can find during seasonally sneezing and all. They will have breathing difficulty when they are inhaled. It leads to severe sneezing. It causes headache. It causes a nausea. It leads to nose blockage or congestion. Continuous sneezing leads to watery eye, itching eyes, itching nose, irritation. And some of the pollen grains, when they come in contact with the skin also, it leads to itching. And when we scratch, it may also lead to burning sensation and development of rashes. So these are certain, you can tell, harmful effect of the pollen grain. So it means, do the pollen grains have only harmful effect? Do they have only harmful effect? Are they not useful also? Of course, some of them are harmful, but at the same time, they also have useful effect. Let's see the useful effect of the pollen grains also now. So pollen grains are also equally useful. See, they have excellent flavonoids. They have a high content of protein. They're excellent source because they're rich source of iron and zinc. And they're also a good source of vitamin B and carotenoids they contain. And they have contain high content of sugar and dietary fibers. See the dietary fibers, carotenoid, vitamin B, iron and zinc, protein content, flavonoids, a substitution food. So besides this, one of the most important one is honey. Honey, they have got lot of useful even in field of medicine like Ayurvedic, homeopathic, they use this. So you can see besides this harmful effect, they are also equally beneficial. Not only this product, even the wax obtained from beehive is also of great use. So these are the beneficial activity, effect of this pollen grains and the bees. Clear? So let's start now. Just a glance of what we have done. Clear? So let's see, this is how the pollen grains are formed in a flower. First, we'll start with the various parts of the flower. See the pollen grain, the beautiful ornament you can see, how it is formed. And this ornamental pollen grains are produced from a flower. Let us take the 4 orals of a flower now. What are the 4 orals of a flower? It starts with the essential as well as the reproductive part of the flower. So this is the different oral. See the stem peduncle. It has this calyx, protective petals, attractive part, followed by the stamen and the carpel. Stamen, carpel, the reproductive part. The other 2 are the accessory part. So anther and filament. When we take a transection or a cross section, you find 4 chambers. These are all the pollen chambers which contain the pollen sac in it. So inside this pollen sac, you have the diploid pollen mother cell, which will undergo meiosis and divide first meiosis 2, followed by the second meiosis 4. Each cell, pollen mother cell, will produce 4 haploid pollen grains or spores. And it is this spores will be present inside the chamber which will undergo mitosis with due to result a large number of haploid 2 nucleate stages are produced. These 2 nuclei will have genetically same content because they are produced from a single cell. And they will form, one will be the vegetative nucleus, the other one is the generative nucleus. So this is a 2-cell stage of the pollen grains. You can see the beautiful ornament with lot of spikes, etc. externally. And internally, this pollen will have the vegetative nucleus and the generative nucleus. So many plants release the pollen grains at the 2-cell stage, but in few plants, again the nucleus internally will divide into 2 pollen bands. So let's see what is happening internal to the pollen or in the transfer section. See, pollen is having an outer membrane exine and a thin inner membrane, the intine. Intine is uniform, but exine is having lot of ornamental outside. It may vary from one species to another species. And this anther will contain a large number of pollen grains produced from the pollen mother cells. So this on maturation, near the connective stomium, it will dehisce because they do not have fibrous tissue, and that will lead to the liberation of the pollen grains to the external environment, thereby releasing the pollen grains. So, hope you all understood what we have did today. Take care. Meet you all in the next session.